Shri Gurubhyo Namaha children. I am happy to welcome you all for our online teaching academy, Ekalaiva. For the class 11, subject biozoology, we are discussing the chapter The Living World. Today we are going to see the topic Taxonomy and Systematics. What is taxonomy? What is the meaning of this taxonomy? Taxonomy is nothing but it is a science of arrangements. Taxonomy is nothing but a science of arrangements of living organisms along with classifications, descriptions, identifications and naming. So all this comes under taxonomy. Taxonomy is a science of arrangement based on the classification, description, identification and naming. Ecosystem of an organisms which includes all flora and fauna. Flora means plants and fauna means animals, including microorganisms in the world. The word taxonomy was first coined by scientist Augustine Pyramus de Candolle in the year 1813. Augustine Pyramus de Candolle. The taxonomy is a theoretical study of classification which with well-defined principles, rules and procedures. Aristotle, very important one. Aristotle is the father of classical taxonomy, traditional taxonomy, classical taxonomy. He is the father. Then Carolus Linnaeus, he is the father of modern taxonomy. So there will be a confusion among Aristotle and Linnaeus. Aristotle is the father of traditional taxonomy and Carolus Linnaeus is the father of modern taxonomy. So children, please note, it is very important. Who is the father of taxonomy? They will never ask. Who is the father of traditional taxonomy and this classical taxonomy, and who is the father of modern taxonomy? Okay. Systematics. Systematics is also called as sequencing. Systematics means sequence. The objectives of taxonomy and systematics are very similar. Both are same. Their goal is to identify. The main goal is to classify organisms with stipulated rules. Okay. So the main aim, the main goal of taxonomy as well as the systematic is to classify organisms based on the rules. Okay. The main criteria of systematics is identifying, describing, naming, arranging and preserving and documenting the organisms. These are all the main criteria of systematics. What are the criteria for systematics? Identifying the organism, then describe the particular organism, naming the organism, arranging the organism, preserving the organism and documenting the organisms. These are the main criteria for systematics. Okay. So apart from the above said criteria, above said features, there have some other evolutionary history of the species and environmental adaptations and interactions and interrelationship between the species are also being investigated in systematics. Apart from these criteria, what are the criteria? Identifying the organism, then describing the organism, naming the organism and arranging the organism and preserving and documentation. Apart from that, there will be another one, that is evolutionary history of the species and the environmental adaptations, how the particular organism adapt with environment, that is environmental adaptations and the interrelationship between the species are also being investigating in the systematics. Okay. Depending on the system of classification, organisms were classified into 2 or 3 kingdoms. So depending on the system of classifications, the total organisms are classified into 2 or 3 kingdoms. Okay. So later it will develop, it will advanced into 4 kingdom classification, 5 kingdom classification, 6, and now it is 7 kingdoms. So organisms are classified into 7 kingdoms now. Okay. So R.H. Whittaker, very important one, in the year 1969 proposed the 5 kingdom classification. So he is the scientist classifying the living organisms into 5 kingdom. What are the 5 kingdom? So he divides the organisms into 5. What are they? The first one is Monera, second one is Protista, then third one is fungi, fourth one is plants, plantae, and fifth one is animalia. So R.H. Whittaker is a scientist in the year 1969 proposed the 5 kingdom classification. So he classified the organisms into 5 major kingdom. One is Monera, Protista, then fungi, plantae, and animalia. So how he classified the organism? Based on the cell structure, mode of nutrition. Cell structure, mode of nutrition, whether it is autotrophs or heterotrophs. Okay. So mode of nutrition, then mode of reproduction, and phylogenetic relationship. So based on the criteria he follow. What are the criteria he follow? Cell structure, mode of nutrition, then mode of reproduction, and then phylogenetic relationship. So based on this criteria, R.H. Whittaker classified the living organisms into 5 kingdom. They are Monera, Protista, fungi, plantae, and animalia. Clear, children? See the tabular column here, classification of living things, living organisms. See, kingdom. Cell type, cell structure, number of cells, mode of nutrition, and examples. These two are said to be Monera, and then Protista, fungi, plantae, and animalia. These are the 5 kingdoms. So cell type, in Monera all the cells are prokaryotic. All organisms are prokaryotic organisms. Prokaryotic means they do not have a well defined nucleus. Apart from this Monera, other than that, Protista, fungi, plantae, and animalia, all these organisms are eukaryotic. They are having well organized nucleus. They are well organized membrane bound cell organelles. Then cell structure. In Monera, the cells having cell wall with made up of peptidoglycans. Here the Protista having the cell wall cellulose. Fungi, cell wall is made up of chitin. In plants, cell wall is made up of cellulose. In animals, there is no cell wall, absence of cell wall. Then we move on to mode of nutrition. In case of kingdom 1 Monera, all organisms are autotrophs, very few are heterotrophs. In case of Protista, autotrophs as well as heterotrophs. In the case of fungi are heterotrophs. They depend on other organisms for the food. Okay. So plants, you know very well, plants are autotrophs, they prepare their own food. Animals, heterotrophs. And then number of cells. In Protista, in Monera, it is unicellular. There is only one cell. Example, bacteria and then cyanobacteria, etc. In case of Protista, most are unicellular and very few, some are multicellular. In fungi, most are multicellular, very few are unicellular, and in plants, all organisms are multicellular. It is made up of many cells. In animals, so number of cells, all are multicellular. Okay. So these are all the major classification of living organisms. R.H. Whittaker classification. He divided the organisms into only 5 kingdom: Monera, Protista, fungi, plantae, and animalia. This table gives a comparative account of different characteristic features of 5 kingdom classification. Clear? Classification has come a long way and now takes into an account even the molecular level, that is DNA and RNA level. So there will be advancement in the classification. The advancement in molecular technique and biochemical assay has led to a new classification that is called 3-domain. So this is a new one, 3-domain classification, 3-domain of life. 3-domain classification was first proposed by Carl Woese in the year 1977 and his co-workers. They classify the organisms into 3 different domains. So they classify organisms based on the difference in 16S rRNA. So based on this criteria, Carl Woese divides the organism, classify the organisms into 3 domains. So based on the difference in 16S rRNA, based on this he classified the organisms into 3 domains. What are the 3 domains? First one is the 3-domain system adds the taxon domain higher than the kingdom. So it is higher than the kingdom. This system emphasizes the separation of prokaryotes into 2, that is bacteria and archaea. So prokaryotic domains, prokaryotes into 2 domains. One is bacteria and archaea. So as I said, 3 domains. First one is bacteria, second one is archaea, and third one is eukarya. These are the 3 domains. Okay. Carl Woese classified the organisms into 3 domains based on the differences in 16S rRNA, based on the difference between these organisms into 3 domains: bacteria, archaea and eukarya. Clear, children? So based on the differences in 16S rRNA, he classified the organisms into 3 different domains. One is bacteria, another one is archaea, and third one is eukarya. How you classify this bacteria and archaea? You just divide the prokaryotes into bacteria one type of organism, and archaea is another type of organism. Okay. Archaea appears to have more in common with the eukarya than the bacteria. So archaea is very common. Okay. So archaea differ from bacteria in cell wall. So archaea is more or less similar to that of the bacteria. So both are prokaryotes. They do not have a well defined or well organized nucleus. So archaea is similar to bacteria, but only one thing, the cell wall is different. So archaea differ from bacteria in cell wall composition and different from the different bacteria and eukaryotes in membrane composition and RNA type. So there will be a difference between the bacteria and archaea. What are the major difference means? Cell wall composition and differs from bacteria and eukaryotes, as well as in membrane composition and rRNA type. Now we see the domain archaea. This domain archaea includes single celled organisms, only one cell, that have prokaryotes which have the ability to grow in extreme conditions like volcano vents, hot springs and polar ice caps. Hence they are also called as extremophils. They can survive, they can grow in the extreme condition. Okay. Then they are capable of synthesizing their own food without sunlight and oxygen by utilizing hydrogen sulfide and other chemicals from the volcano vents. So all archaea are autotrophs. They can prepare their own food. Instead of taking the sunlight and oxygen, they prepare their food by utilizing the hydrogen sulfide and other chemical from the volcano vents. Okay. So some of them produced methane, few living in salty environments, and other thermophils which thrive in acidic environments and in high temperature. Then we move on to domain bacteria. Bacteria, you know very well, it is a prokaryotic organism. The cells have no definite nucleus, and then DNA exists as a circular form. Bacteria having the circular DNA, circular chromosomes, and do not have histone associated with the DNA. So, circular type of DNA, there is no histone present in the DNA. Okay. So, they do not possess membrane bound organelles except for ribosome 70S. Other organisms there is no membrane at all. So, without membrane. The cell wall contains peptidoglycans, many are decomposers, some are photosynthetic, and very few are disease causing. Okay, very few causes diseases. They are beneficial probiotic bacteria and harmful pathogenic bacteria which are diversely populated. Then cyanobacteria. Cyanobacteria are photosynthetic blue-green algae which produce oxygen. They play a key role in the change in the atmospheric oxygen levels from anaerobic to aerobic conditions. Then we move on to domain eukarya or eukaryotes. Eukaryotes is not mean that they have well organized nucleus. So eukaryotes are animals which have true nucleus and membrane bound organelles. DNA in the nucleus is arranged as a linear chromosomes. So inside that nucleus the DNA are in the linear chromosomes with histone proteins, ribosomes of 80S type in the cytosol and 70S type in the chloroplast and mitochondria. Organisms in this domain, in this eukaryotes, classified under kingdom namely Protista, fungi, plantae and animalia. In 1987, Cavalier-Smith revised the 6 kingdom system to 7 kingdom system. Okay. So the concept of super kingdom was introduced and revised to 7 kingdom classification. The classification is divided into 2 super kingdoms. One is prokaryota, another one is eukaryota, and 7 kingdoms, 2 prokaryotic kingdoms, eubacteria and archaebacteria, and 5 eukaryotic kingdoms, protozoa, chromista, fungi, plantae and animalia. Thank you children. To receive our online lessons, please press subscribe button and you will receive the latest updates. Thank you.